Okay, so it's a great pleasure for me to introduce Professor Megan Morrison from the College of Computing at Illinois Institute of Technology. Professor Morrison is an applied mathematician and I would say mathematical biologist. She works at the intersection of data science and dynamical systems. With particular focus on extracting lower dimensional representation on the dynamics of the neural systems. And today she's going to talk about her work on C. elegans. So thank you very much, Professor Morrison, for being here.
Yeah, thank you so much for having me. And I'm excited to talk about my work as a part of your seminar series. So today I'm going to be talking about reproducing C. elegans premotor neural activity, neural dynamics, and decisions with a biophysical network model. And this is work that I did with my postdoc mentor, Lai Seng Yang, who's at the Quran Institute. So first off, I'm going to give some background about what are C. elegans, what does their nervous system look like, and what behaviors they exhibit, and then how we constructed a parsimonious network model for locomotion decisions, and then talk about what we did for our model simulation and analysis, and then conclude with some future directions. First off, what are C. elegans? They're these very small nematodes. They're only about 1 millimeter in length, and they have very few neurons. They live in the soil, and they move by contracting their muscles. And experimentalists really like them because they have basic behaviors. It's relatively easy to measure the neurons, first off, because there's not that many of them, and they can actually ID label the neurons as they're firing or like changing in voltage and as the C. elegans is producing different behaviors. So here are some of the basic behaviors it exhibits. It swims forward, it can do a couple different types of turns, and then it usually does a reversal before it executes a turn. And so these basic behaviors then are scaffolded together in more complex behaviors where it changes between basic behaviors to, for example, forage for food, escape predators. They can follow gradients that maybe lead them to a food source. And they also like to form social aggregates. Here's an example of an experiment where they put a worm on this plate that had food, and then they could watch the worm over time as it tried to track down the E. coli patch. And so it spent a lot of time here, kind of turning around, exploring this area, and then eventually the worm decides to leave a certain region and go explore a different area. And so what we want to understand is how the worm generates these basic behaviors and how it decides when to switch between different behaviors for example, allowing it to forage for food. Here's a video of some C. elegans that have been lit up with fluorescent proteins, and you can see some of them have formed social aggregates. Some of them are kind of exploring the region. And you can see that they exhibit these basic behaviors where they swim forward, they'll stop, reverse, turn, and then they'll go into quiescence once they found a food patch or formed a social aggregate. So this is what the behavior time series looks like, and we want to understand when these switches occur between basic behaviors. So how does the nervous system produce stochastic switching between different states, and why is that happening? So first off, here's a little bit more about the actual nervous system. There's sensory neurons, that's like the first layer of neurons, and they can sense a lot of different things like UV light, temperature, oxygen, etc. There's about 90 sensory neurons, and then the next layer is the interneurons. These neurons integrate the information that they're getting from the sensory environment, as well as send signals to the motor neurons, which are primarily responsible for coordinating the muscle contractions to allow the C. elegans to move around. It looks, the way that I made this picture, it looks feed forward, but actually these neurons are highly recurrent. So for example, motor neurons are even connected back to the sensory neurons. There's no very clear flow of information from the beginning to the end. There's lots of recurrence. A subset of the interneurons have been identified as premotor neurons. And those experimentalists have honed in on as making the decisions about locomotion. So here's what the actual connectome looks like, or at least part of it. This isn't all 302 neurons, but these are, I've labeled some of the premotor neurons and then neurons directly connected to it. And you can see that even like one step away from the premotor neurons, the network gets pretty large pretty quickly. Lots of recurrent connections. There's also two sort of different types of connections. There's gap junctions, which kind of average the voltage activity between the neurons. And then there's synapses, and those are directed, those are chemical connections. So what data do we have available? First off, there's the anatomy data. So we know what the connectome is. All the neurons are connected in exactly the same way from worm to worm, which is great. So then when the experimentals start doing different experiments, they know they're operating on the relatively the same network every time. Then there's the physiological data, so they can measure from single neurons using a voltage clamp, or they can measure from multiple neurons and get this whole brain imaging data, which is what I mostly used to fit the model. And then behavior, so time series of basic and complex behaviors, as well as when they ablate different neurons, they can see how the behavior changes and then infer the importance of a neuron to a behavior that way. So we want to build a model that will incorporate lots of different types of data that's available to us. So going back to the connectome, the first question is how to isolate and model one part of the central nervous system. So we're not having to model everything, but just the neurons and the connections that are directly relevant to these locomotion switches. So here's our conceptualization based on the literature. There's some subset of neurons that have been identified as associated with locomotion. Then a subset of them have been associated with forward motion, another subset associated with reversal. There's a regulatory subset, and then those all interact with the motor neurons. So our objective is first to select a subsystem that will reproduce the main behaviors that we're seeing. So our plan was then to extract a subset of nodes and edges that can produce the stochastic switching, because the stochastic switching is what's underlying the search behavior. One of the main challenges, it's unclear which neurons belong in a model and which are not relevant to this locomotion. There's also different neurons in different data sets. So although we do have multiple whole brain imaging data sets, not all of the neurons are labeled in each data set, which makes it a challenge to take multiple partial data sets and use them all together to fit a model. So here's the model we came up with. Here's our final set of model neurons that we got from first taking some seed neurons that are highly relevant for driving forward or reversal, and then we iteratively built out a subnetwork from those neurons until we got a final set whose overall dynamics would produce stochastic switching. So here, our final set of model neurons has forward driving neurons and reversal driving neurons. And then what we did is we took the model neurons and we simulated their activity, the model neuron activity, in the presence of signal input from the remainder of the network. So from presynaptic neurons as well as presynaptic sensory neurons. And then those neurons in turn drive the motor neurons. This wasn't part of our model. We didn't simulate the muscle activity. We just assumed that with our model and the production of like activity for the forward and reversal neurons, that would then feed downstream to the motor neurons. But other mathematicians have worked on modeling muscle activity. So that's something maybe in the future we could incorporate into a model. So here's the ODE that we use to simulate the neural activity. This is the dynamics for each model neuron. It has a few different components. First off, it has the intrinsic dynamics. So that is what the neuron is going to do without having any input fed to it from other neurons. So its intrinsic dynamics are nonlinear. We see that from voltage clamp data, where we can see that the neuron will respond non-linearly to different levels of voltage. It's not just linear. And so, although we have a similar shape for the intrinsic dynamics of each neuron, we do fit the F function depending on the neuron. Our next component is gap junctions. These are electrical connections, and they're symmetric. So if two neurons are connected with a gap junction, the higher voltage neuron will kind of drag up the lower voltage neuron until they average out. And then we have synaptic connections. And so these are directed and they have a sign associated with them. Although we know the location of chemical connections, we don't actually know the weight or the sign that is associated with them. And so that's something that we got from our regression analysis. So first off, the intrinsic dynamics. We model the intrinsic dynamics with a cubic function with an individualized bias term, DI. We get the approximate shape from voltage clamp data, which measures how the cell is responding to various levels of voltage input. And we find that a lot of these neurons, looking at the voltage clamp data, have multiple stable states. They have a low excitatory state, which is stable, and then a higher voltage state that is also stable. And then by perturbing the voltage, the cell can be pushed between the low activity and high activity state. So here's an example
And there
voltage
might
clamp.
be a bifurcation as well by moving the DI, right?
Yes, exactly. You could, by changing the bias parameter, you could induce a saddle node bifurcation in either direction. Um, so here is some example calcium imaging time series, and it kind of correlates or responds to the voltage clamp data that we're seeing, in that the activity of each neuron tends to either be at a high voltage state or a low voltage state, and then will fluctuate between those two states, depending on what the rest of the network is doing. So that's why we decided to approximate the intrinsic dynamics with this cubic function with a bias term. So our next term in our dynamics is the connectome data, which I talked about. It's kind of the averaging of the voltage of two neurons. And these weights we can read off from the connectome data that the experimentalists have collected. And then the last component is the synaptic weights. And so after determining the intrinsic dynamics, the gap junctions, we actually determine these synaptic connection weights and signs as well as the bias term DI at the same time. And so just focusing on this term, here we take the connectome. It will determine the synapse locations, but doesn't give the weights and signs of the AIJs. And so what we do is we do regression for these weights using 22 different whole brain imaging data sets, which are on different worms, different experimental conditions, and different neurons measured. And so we do a lot of different regressions, and then we average over all the regressions to get a general set of weights that isn't specific to any individual worm, but will generically describe, hopefully, any given worm. Not perfectly, though. So here's an example time series where a lot of the neurons have been identified. And what we want our model to do is to be able to reproduce this type of activity. So I'd like to mention the whole brain imaging experimentalists who have done a lot of work to collect this data, which makes the modeling possible, because none of this is possible without really high quality data sets. So the first result that we get is we get synaptic weights and signs, and that allows us to then further analyze the network structure and use that to understand how does that contribute to the dynamics. So the largest structure that we see between the forward and reversal neurons is inhibition between the two clusters, and then excitation within each cluster. So the dominant structure is this excitation within, inhibition between. And this goes along with some previous work where they hypothesized using Markov models for stochastic switching that this type of network structure could be the reason why they see this Markov model switching. So they didn't actually model the network itself. They modeled the behavioral output, and then they thought about what types of structures could actually give them this behavior. We next look at the connections from the presynaptic neurons to the model neurons. And so we kind of ranked them from forward promoting to reversal promoting. So some of the neurons, especially those at the two extreme ends, they definitely form stronger connections with one subcluster versus the other. So we can hypothesize that they're important for promoting one type of behavior, swimming forward or reversing, versus a lot of the neurons in the middle, they form both positive and negative connections with both clusters. So it's unclear whether or not they're regulating the locomotion behavior or they just have some other function altogether, and they're not really involved in regulating locomotion. So here's the general structure that we see: forward and reversal clusters, and then subgroups of either forward promoting neurons or reversal promoting neurons. So here's what our network does when we simulate the activity of like the 15-dimensional nonlinear ODE that we built under the influence of signal input. So the top plot here, this is showing the calcium imaging data from a particular data set. And then we take the signal input from the same data set, feed it into our model, and this is what we get for our simulation time series. So we're able to reproduce some of the stochastic switching between forward and reversal neurons. So in this case, when the forward neurons, the blue neurons, are more active, that corresponds to forward decision. The worm is deciding to swim forward, and then when the reversal neurons are more active, that corresponds to it's deciding to reverse. So we can dimension reduce this down to just which cluster is more active, forward and reversal, and then get a behavior time series from this. So the top is what's happening in the data. The bottom is what's happening in the simulation. So some of the switches happen accurately and at the appropriate time. Other switches, they're delayed, or maybe they're not happening at the correct time. And so we can further analyze why certain switches aren't occurring in our model. So this is one example of one data set that we reproduced, but we also did this with a lot of the other data sets. And then we wanted to understand how well is our model in general fitting these different data sets. So we did that in a few different ways. So first off, we looked at the correlation between the neurons. So we looked at the correlations that we saw in the data and then compared that to the correlations we saw in our simulations to see, are neurons, when they're correlated in the data, do they tend to also be correlated in our simulations? So that's one way that we can get at how well our model is like reproducing these higher order statistics. So in our model, the correlation matrices are similar. We also see like forward neurons are highly correlated, reversal neurons are highly correlated, and then they tend to be anti-correlated to each other. Another way to get at if our simulation is matching the data is looking at the probability distribution functions of individual neurons. So for example, these are the PDFs of some of the forward neurons. So it just tells you about how much time is it spending in different states. And there's two humps here that kind of correspond to neurons tend to either be in the low activity state or the high activity state, and then switch between them. In our simulation, we see something similar. However, in our simulation, the PDFs don't exactly match what we see in the data. For example, the neurons in our simulation spend a lot more time in the low activity state and not quite as much time in the high activity state.
So I have a question on this. So your matrix W that you have in your model. So that's basically act linearly on xj minus xi, right? Or is that yeah, acts linearly? And and is it symmetric?
Yes, it's symmetric and it is linear. There's no nonlinear term, nonlinear function that the activity is being put through.
Got it. Thank you.
Yeah. Okay, so something else that we wanted to look at is are different long-term behaviors being represented by our model. So here's like two extremes. One is this frequent switching that we see in this data set, where you can see it doesn't spend a whole lot of time in either the forward or reversal state. It switches pretty quickly. And this corresponds to dwelling behavior, where the C. elegans will kind of explore a smaller space. So here's the calcium imaging, here's our simulation, and we do see this like high frequency switching in our simulation. And this isn't because our model is different for this data set, it's just receiving different input signals from this specific data set, which then we can use to answer the question like, why is this frequent switching occurring? What in the input signals is causing this to happen? And then the behavior. On the other end of the spectrum, you have this roaming behavior. And roaming behavior, you see very long stretches of either forward or reversal activity. And this will correspond to when a worm is trying to escape a region and go somewhere else. And so in our simulation, we do see roaming-like behavior. We do see longer switches of either reversal or forward behavior. However, the switches aren't matching as well as they were in some of the other cases. But the qualitative overall behavior is the same. Another result that we got is looking at the contribution of gap junctions versus synaptic connections and what is like the function of these two types of connections. Why does the worm have both and not just one? So for example, here's a forward neuron cluster just showing the gap junctions, and you can see that they're like pretty well connected by gap junctions. Here's an activity of just the forward neurons, not the reversal. So what we did in this like experiment on our simulation is that we cut either the gap junctions or the synaptic connections, and we saw what qualitative behavior would come out of that. So cutting the synaptic connections and only having gap junctions, we see the neurons stay highly correlated. This kind of makes sense because gap junctions keep neurons correlated, but you don't see the up and down switching occurring as much as in the data set. On the other hand, when we instead cut the gap junctions but keep the synapses, you see much more like dramatic switching. However, the neurons are now no longer correlated as well as they were. And then you put them together. So this is the original experiment where we have all of the connections that should be there. And you get the correlation and the switching. So this kind of indicated to us that the function of gap junctions may be to coordinate activity, and then the function of synapses is to instigate switching between behavioral states. And then we saw a very similar phenomenon looking at the reversal cluster. So here's the data. Here's the simulation with only gap junctions, simulation with only synapses, and then put them together, you get higher correlation and as well as the switching activity. Something else that we wanted to use our model to do is try and understand the purpose of different sensory neurons. What are they contributing to the dynamics? And so we artificially turned on different sensory neurons and put that input into the model to see how the model activity would change when we artificially turn on or off a sensory neuron. So we did it heavy side input to the model, which is not necessarily very realistic because neurons don't just like turn on and stay on for long amounts of time like this. But we just wanted to see if there were any dramatic changes before and after turning the neuron on. So here's an example data set and what we get with like no pulse to the model. And then this is what happens when we apply a pulse to the model through the ASH neuron. And so this neuron, it is important for an escape response. Experimentalists have identified that when this neuron turns on, then that will like promote the C. elegans escaping by swimming backwards very quickly. And we see that in our model, when we do give this pulse through the ASH neuron, it promotes a reversal behavior, which is kind of what we were expecting. Here's another famous neuron, OLQ. This is supposed to promote foraging, and when we give the model a pulse through this neuron, it turns on the forward neurons, biases the network towards forward motion. So these are two examples of neurons whose function has kind of already been identified, but we would like to do the same thing for a lot of the other neurons that are maybe not as well studied to try and understand if they contribute, how they contribute to locomotion, and if they bias the network in any particular way. So our model isn't the only model where we're simulating C. elegans neural activity. There's a lot of different models that contain different features. So here are some different features that these types of models can contain. First off, they can either be based in biology or be more abstract. They can include the connectome data, so be constrained by the connectome. Some models, they are fitting their model to whole brain imaging data. Others aren't. They're using other types of data. And then some models, they either have stochastic switching dynamics occur in their model, or that's not something they're trying to reproduce. And then some models are trying to look at stochastic behavioral sequences. So looking at the previous models, like some of them are biophysical, where they're actually looking at the connections that actually exist in the network. And then another set of models is data-driven. And so they're actually using a lot of these data sets to fit parameters in their model. So a subset of these is phenomenological, which means that they're reproducing a phenomena that they actually see in the network. So the stochastic switching. And so our model, the goal of our model was to have it be based in biology, use these connectome constraints, fit it to whole brain imaging data, but then also be able to reproduce like the stochastic switching that we see. So some of the challenges that are prevalent in math biology, but definitely showed up in this modeling attempt, is to build data-driven models with data sets that are disparate, partial, and descriptive. and then create models that go beyond just reproducing phenomena under a set of constraints. So some of the challenges that we ran into, first off, extracting a relevant subnetwork. The way that we approached this was to iteratively build subnetworks and then test their representative ability. We wanted to incorporate diverse data sets into a single model. So our strategy was to select variables and model forms that can be fit given a set of data sets. And then we wanted to derive governing principles from our model. So we approached this by doing dimension reduction and then analyzing contributions to the low dimensional activity. And this work, it's on archive at this location. Thank you so much.
Awesome. Thank you, Professor Morrison, for the great presentation. So I have a few questions myself.
Yeah.
So I'm gonna ask them, random order. So the first one is, so you talked about this stochastic switching. So
Mm-hmm.
do you have, is there some random term in the equations that you use? Or is it more something like that it's stochastic in the sense that it's unpredictable? Or what do you mean with stochastic switching? That's probably the best question. Because you know, in my
Yeah.
understanding of the equations, there are there are only deterministic components, if I'm not mistaken.
Yeah, there are only deterministic components. And so, yeah, there's no stochastic terms here. It's stochastic because you have different inputs that are coming in from the presynaptic neurons and from the gap junctions with the presynaptic neurons. And so, yeah, it's a deterministic model, but because like the presynaptic neurons, they're noisy and you have different inputs depending on the data set, the output ends up looking stochastic, but it's not, according to our model, it's not technically stochastic.
Got it. So also on this matrix here. So have you tried to change the gap junctions component, make them nonlinear, like just sign instead of Wij? So having sine or cosine nonlinearity. So like let's say sine of xj minus xi with a coefficient which depends on I and j. Have you tried the model like that?
So for the W's, those don't have signs because it's just a
Sorry, I mean oscillatory nonlinearity, cosine or sine or something like that, like something like a sinusoidal wave.
Oh, okay. Oh, because our nonlinearity here, it's the ReLU function. It's not sine or cosine. So we represented it as the ReLU function because we thought that was the simplest thing to do to introduce a nonlinearity because presynaptic neurons, if they're like very like hyperpolarized, if the voltage is low, they're not affecting the downstream neurons. So it's kind of zero effect. And then once they have a higher voltage, then they are affecting the downstream
Oh,
neurons.
so you were that is for the voltage term, for the intrinsic dynamics term, correct? While can you make nonlinear the gap junction, so the electrical component, maybe with a sinusoidal nonlinearity?
Oh, yeah, that we could add that in. Yeah, so our model is kind of minimally parameterized. So it doesn't like we could add features like that that would make it more realistic. Um, yeah, so that's a good point is that it may not technically be linear and it may be better to add a nonlinearity on there as well.
So the other questions on this equation is, if I remember correctly, you average for estimating the A's, you do some averages, right? So now, what I'm wondering there is, how do you do the average? Because it can be like a simple arithmetic average, or it can be a little bit more complicated average, because that component there might have some correlation, since you use regression, right? So it might be that there is some correlation component there, which is basically almost an angular variable. I don't know how to say that. And because of that, it may be that if the average is done in different types of methods, you might have different behaviors emerging. Like in angles, if you average two angles, you may get with arithmetic, usual arithmetic average, you may get that you have some cancellations. So, and in fact, it seems to me that at certain point, so that's the question, right? Do you do simply arithmetic average, or do something a little bit more complicated? Because depending on that, you might get some slightly different outcome.
Yeah, no, that's a good point. What we're doing right now is just a basic average, just arithmetic average. However, this isn't like the most ideal way to get the general weight. So what we're doing now is kind of going back and trying to come up with a better, more rigorous way to come up with these general rates rather than just doing a bunch of regressions and then averaging them.
Awesome, awesome. So the other thing is so at the beginning you talk about different behaviors, right? That emerge as basic behaviors, I think you call them. And one of the things that I was wondering is this. So sometimes when you have some differential equations or some maybe complicated models, you have some simpler solutions, like traveling waves. So sometimes, I don't know, you have a bump which travels with constant velocity or something like that. And I was wondering if in the model that you have, you can actually construct some of these solutions explicitly. For example, you have you have something which is varies on time as translation or something like that. So that this way you have just simply a simple term appearing on the left hand side, maybe derivative, and then you try to solve it explicitly, at least in a low dimensional case. So I was wondering if you tried something like that.
So we haven't tried to come up with explicit solutions. We've just been doing like numerical simulations
Yeah,
of
it
these.
could be, it could be even that, you need at the end some numerics, but you have that the model itself, you can show relatively that the model itself admits something like traveling waves, something like traveling waves. I'm not saying traveling waves because there is not a spatial component in the model, but it could be traveling in the sense that the signal moves from neurons to neurons in a simpler way in some parameter settings.
Oh, gotcha. Yeah, no, that's a good point. We should look into that. Yeah, that's not something
It
that
might be
I've tried
complicated
to
because
do
it
with the
might
model.
depend on this function f and some other coefficients there, but the linear component might be, help a little bit, but maybe that in with just when you try to simulate, when you try to study just a couple of neurons, maybe you reduce to one single equations, and then you can pick up some interesting dynamics. Yeah, yeah. And and maybe my last question. So I was wondering this, like now is there so the data sets that you're using are kind of complex in the sense that, as you explained, there are many components there. Something comes from the voltage, some other comes from connectome. You have the synaptics connections and something like that. So you have many different components, right? So C elegans is a relatively simple system because but for more complex systems, are there all these data sets available? Like, is there something that is for more complex systems? Because this is already hard, I believe, because to have all this information at the same time, it's already complicated. But is there something for more complex systems than the C elegans?
Oh, like if there's whole brain imaging,
All brain imaging, all synaptic connections, all the voltage information. Is there something that is for more complex systems?
I know for like Drosophila, they just finished coming out with the connectome for Drosophilus, and they have like thousands of neurons, so a lot more than worms. I'm not sure if they're not really at the point where they have whole brain imaging data. So there's kind of a trade-off. Like if there's too many neurons, maybe they can measure like the entire space, but they can maybe get cell to cell resolution. Versus the elegans, because there aren't that many neurons, they can actually look at like what each individual neuron is doing rather than a region. But for some models, maybe it's not important to get like that cellular resolution. Maybe you want just a model that represents what's happening in a particular brain region. And so
Yeah, awesome.
maybe
Yeah,
having that detailed.
there is nothing like this for a dog, right?
Oh yeah, no, definitely not. Like there is like lots of brain information, like maybe EEG or fMRI data, but it's not, you can't see what each individual neuron is doing. And when you get to more complex organisms, like they have different numbers of neurons, they're connected in a different way, and they're more individualized versus C. elegans are so simple that they're the same across each worm. And so then you can more easily just build a single model that will represent all of them.
I said
Uh,
simple too, but of course, also here there are so many things that one has to understand that it's very interesting. And
yeah.
I think thanks a lot for your time and also for taking extra time to answer questions. And it was great, a very interesting topic. And so thank you very much for taking time to present in our seminar series.
Yeah, thank you so much for having me.
My pleasure, my pleasure. So I'm gonna stop the recording now.